blood to the lungs? Or where does blood go when it leaves the right atrium, the right ventricle? Or where does blood first enter when it's coming back from the lungs? That's the left atrium. Things like that. So you definitely have to know this pathway of the blood. You should also know, the names of the blood vessels. You should know whether the blood traveling through these blood vessels is oxygen poor or oxygen rich. Does it have a lot of oxygen in it or not? Okay. How you doing, Kelsey? Not at all? Okay. Now the rest of the pathway, of course, is our systemic circuit. We start where we left off here with oxygen-rich blood in the left atrium being pumped into the left ventricle, that big heart chamber. The left ventricle pumps oxygen-rich blood out to the body through the aorta. The aorta branches into smaller and smaller arteries and arteries that serve specific organs and limbs and your big toe and what have you. Capillary exchange occurs. This is what we talked about at the beginning of class here, where we drop off the oxygen, we pick up the CO2 waste, and now we have oxygen-poor tissue, which is going to be flowing back toward the heart through larger and larger veins until eventually we get to the vena cava. And we've got the superior vena cava coming down from your head and arms, and the inferior vena cava actually coming up from the lower part of your body. And all that blood eventually empties into your right atrium. One quick thing I want to remind you of. Last time we talked about difference between arteries and veins, right? Arteries take blood away from the heart. Veins bring blood back to the heart. Most of the arteries in your body carry oxygen-rich blood. So the aorta and the other arteries that go off to your different various organs have oxygen-rich blood in them. And a lot of people kind of have the impression that all arteries have oxygen-rich blood in them. What's an exception to that rule? Which artery actually has oxygen-poor blood in it? It's in the picture. Yeah, I heard somebody say it. The pulmonary artery. The pulmonary artery actually has oxygen-poor blood in it because it's taking the blood from the right side of the heart, hasn't made it to the lungs yet to pick up more oxygen. So this is the one artery in your body that doesn't have oxygen-rich blood in it. Most do, this one doesn't. Okay. Now, a lot of people also have the impression that veins have oxygen-poor blood in it. Most of them do. Most of the veins bringing blood back from all of your tissues have oxygen-poor blood in them. That's true of the vena cava. There's one exception to that rule. Which of your veins has oxygen-rich blood in it instead? The pulmonary again. The pulmonary vein coming back from the lungs has loads of oxygen in it because it just passed through the lungs and picked up oxygen. Okay? So keep that in mind. The pulmonary artery and vein are kind of the opposite of everything else in your body as far as containing oxygen or not containing oxygen. That's one thing where people kind of get stuck sometimes. Too much left to go here, but I'm going to talk a little bit about how this process is controlled. How do we move the blood through? How do we operate this pump? Well, your heartbeat, you've probably all listened to your heartbeat before. Maybe you've had a stethoscope in some class in grade school. You get to listen to your heartbeat. You probably have an idea of what it sounds like. But each heartbeat is referred to as a cardiac cycle. And it has two basic parts to it. First of all, the systole part, which is when the heart muscle is contracting. And then there's a period when the heart is relaxing afterwards, called diastole. So you've got contraction of the heart followed by relaxation, followed by contraction again, followed by relaxation. Except it's not that simple, because the heart actually contracts in two steps. The whole heart doesn't contract all at once. Part of it contracts, then another part of it contracts, and then it all relaxes. Okay? So the contraction of the atria, the entranceways to the heart, isn't simultaneous with the contraction of the ventricles, the exits to the heart. The atria contract first, followed a split second later, just a couple tenths of a second, by the ventricles. The reason for that is the atria have to squeeze first and squeeze blood into the ventricles. Then the ventricles squeeze to squeeze blood out to the body or the lungs. So if they both squeezed at the same time, the ventricles wouldn't be very effective. They have to first fill up with blood, then they can squeeze it out to the body. So your body has to have a way to regulate that to get part of the heart to contract separate from the other half of the heart. And also as it's contracting, that's one of the reasons why we have these valves, these AV valves and semilunar valves that are going to be preventing backflow of blood. Because as the ventricles contract, we only want blood to be going out through the blood vessels, not back up into the atria again. So these AV valves will prevent blood from going from ventricles to atria, or prevent blood from going from these blood vessels back into the ventricles again. So here's how the cardiac cycle works, all right? First, the atria contract. So you can see contracting atria in this picture here. That squeezes blood into the ventricles. Then the ventricles contract. And as the ventricles are contracting, we'll go back to this picture, notice that the AV valves are open to allow blood to flow from the atria into the ventricles. But when the ventricles are contracting, those valves are closed. So blood can't go back into the atria. It can only go out through the attached blood vessels. Then both chambers of the heart, or all four chambers of the heart, are relaxed, and the heart will start filling up with blood again. The atria start filling up with blood. Then we repeat that. The atria contract, squeezing blood into the ventricles. Ventricles contract, squeezing blood out into the blood vessels, to the body, to the lungs. Then both of them relax. And the whole thing only takes about 1 second or so if you have a heart rate of about 1 beat per minute. Could be a little bit faster if you have a faster heart rate. If you've ever listened to your heartbeat with a stethoscope, you hear kind of a lub-dub. What you're actually hearing is the closing of the valves. So as the ventricles contract, the AV valves close, that's the lub. And as everything relaxes, the semilunar valves close, and that's the dub. So you're actually hearing the valves closing after the blood has gone out of the chambers. Now, the way that we get the different parts of the heart to contract at different times, we have two tissues called nodes in our heart. They have both muscle-like and nerve-like qualities. They can pick up signals from your nervous system coming from your brain. They're under control of your brain, for example, but there's two of them in your heart. One is called the SA node, or sinoatrial. That's because it's in one of the atria. It's actually, on my little drawing here, approximately right up here is where you would find the SA node. The job of the SA node is to initiate the heartbeat. It actually is going to make the atria contract. So the SA node is sometimes called the pacemaker of the heart. It picks up the signal coming from your brain telling your heart to contract, and it first causes the atria to contract simultaneously together. So the SA node initiates the heartbeat. Then there's a second nodal tissue in here, which is called the AV node, or atrioventricular. I bet you could guess from that word that the AV node is halfway in between an atrium and a ventricle. And in fact, the AV node is approximately right in here. Okay? So the AV node is right in between the right atrium and the right ventricle. So once the SA node has picked up the signal coming from your nervous system, it makes the atria contract, and it sends out a signal at the same time. That signal is then picked up by the AV node, and the AV node is then going to cause the ventricles to contract a couple tenths of a second later. So the SA node makes the atria contract, blood squeezes into the ventricles. Then the AV node causes the ventricles to contract, blood squeezes out into the blood vessels. Now there are some special fibers that are in your myocardium, in your heart muscle, called Purkinje fibers. I'll show you a picture in just a minute. I always think that's a cool word, Purkinje. Purkinje fibers help to transmit the signal down to the thick walls of the ventricles down here. So they help carry that signal. And actually, it causes the bottom of the ventricles to contract slightly before the top of the ventricles, because remember, in the real anatomically correct heart, it's squeezing the blood upwards through the blood vessels rather than downwards. So it actually causes the ventricle to contract kind of in an odd way. So here's a look at the more anatomically correct heart again. And here's our SA node up here in the right atrium. That's the pacemaker of the heart. It receives a signal from the brain through your nervous system. And that signal spreads out through the myocardium up here in the atrium, causes them to contract. That once that signal reaches the AV node, that's right here in between the right atrium and ventricle, it travels through these fibers. The AV bundle and the Purkinje fibers are out here, helps transmit that signal to the ventricles and gets them to contract second. So that makes sure that the heart contracts in this coordinated way. Atria first, followed by ventricles, followed by a period of rest. Then the atria again, then the ventricles, then another period of rest, all taking about a second or so. Now, what controls the AV node, or the SA node? Sorry, not the AV node, the SA node. What controls the SA node? Well, there's actually some hormones that your body produces, and a part of your brain that's devoted to controlling your heartbeat. So first of all, you have part of the reason why you might have to control your heartbeat and the strength of your heart contractions is to change your blood pressure, get your blood pressure up or down a little bit. So you have some baroreceptors, as in like barometric pressure, right? There's some receptors in your aorta and your carotid arteries, the ones that are up here in your neck, that monitor your blood pressure. If your blood pressure's too high, these little receptors get stretched a little too much, and they say to the brain, whoa, blood pressure's too high, we need to stop pushing the heart so hard because it's pushing the blood out too strongly. There is a cardiac control center in your medulla oblongata. Remember that your medulla also controls things like respiration rate. Your medulla controls things like peristalsis in your digestive system to push that food through. So this is the automatic or the autonomic part of your body functions. So your medulla oblongata cardiac control center is able to alter your heartbeat, slow it down, decrease the amount of force of your heart contractions if your blood pressure is too high. Let's say that you're busy running a race, you're going to need more oxygen getting out to your tissues. It's going to speed up your heartbeat so that you can deliver more oxygen out to your tissues. So it can both speed up and slow down your heartbeat. You also have some hormones produced in your adrenal glands. Those are those glands that sit back here on top of your kidneys. So the adrenal medulla, the middle section of your adrenal glands, produces a couple hormones, epinephrine, which we sometimes call adrenaline, or norepinephrine, both of which can stimulate the heart. These help with that fight or flight kind of response. Have you ever had somebody jump out and scare you, and you're like sitting there with your heart pounding and racing all of a sudden? That's because you release these hormones and they stimulate your heart to start speeding up, just in case you need to punch somebody in the face or run away or something like that. We're almost done. I feel like I should be singing or something to entertain you during these little breaks. I don't know any biology songs. Good to go, Kelsey? Okay. How do we measure the heartbeat? Well, some of you are getting a chance in lab to do this week. We're, well, not ECGs, but doing things like blood pressure, for example, taking your pulse. Has anybody had an ECG or EKG before? So describe it to us briefly. What did it look like when you were getting an ECG? Right, so you guys have probably seen this in like those hospital shows like ER or Grey's Anatomy or something like that. But they hook up these electrodes to your heart. And what they're actually trying to measure with the ECG is the electrical signals that are being transmitted when your heart is contracting. So as your atria contract, that sends out an electrical signal. As your ventricles contract, that's sending out an electrical signal. And then as those heart chambers are resetting themselves back to their resting position, they also send out an electrical signal that can be picked up by these electrodes that you have stuck to your chest. So here's a typical picture of what a single ECG trace looks like. And it has three little bumps to it. One little bump here, big bump here, and another bump here. So they name these little bumps. This is called the P wave. And the P wave is the electrical signal sent out when your atria contract. It's a pretty weak little wave here. The QRS wave, the big one, that's the electrical signal sent when the ventricles contract. And then the T wave is the electrical signal sent as the ventricles are resetting themselves back to resting position. Now, there is a wave in there somewhere when the atria are resetting themselves back to resting, but it's kind of lost in here somewhere. We can't really see it as a separate wave, okay? And so you're able to look at an ECG reading. Here's somebody hooked up to an ECG. The machine looks just like that, pretty much. Yeah. So here's somebody hooked up to an ECG. This is a fairly normal ECG reading. So a doctor could look at this and see whether these waves all looked correct. They could see whether your heartbeat was beating regularly, for example. Or they could see if something is very, very wrong. And this is what we call ventricular fibrillation. The ventricles are basically just going blah, 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 and pumping the blood without waiting to fill up with blood. They're just sort of squeezing out of control. And that sends out a whole lot of electrical signals, but doesn't do your body any good because you're probably not being very efficient at pumping blood out if you don't wait for the ventricles to fill up with blood before you squeeze. So usually ventricular fibrillation, that's when they're on the doctor shows and they're like, he's in defib, get the, what are those little shock things called? Huh? Yeah, defibrillators, that makes sense, doesn't it? So that's when they shock them, and they basically temporarily stop the heart and hope that it restarts itself in a normal rhythm. So that's what it would look like if somebody needed to be shocked. All right. Now, you can also pretty easily measure your pulse, and that's because your arteries are stretchy. Remember that they have a layer of muscle around them, and so when blood flows through the arteries, pushed out by the heart, your arteries stretch a little bit. So good places to take your pulse. You can feel it in your wrist. You can also pretty easily feel your pulse in your carotid artery here in your neck. You've probably all had to do that for a PE class or something like that before. You can also measure your blood pressure. You can actually figure out the pressure being exerted on the walls of your arteries. And blood pressure actually has two numbers to it. The upper number is the systolic pressure, which is the amount of pressure that's being put on those blood vessels when the ventricles are actually contracting. They're squeezing, they're pushing the blood into the arteries, and that's putting a fairly high pressure on the arteries. The lower number on the bottom is the diastolic pressure. That is the remaining pressure that's still in the lungs, in the blood vessels, in the arteries, when the ventricles are relaxing and not actively pushing the blood out. So what's a typical blood pressure reading? What would it be if you're healthy? What was that? Yeah, like 120/80 is a usual healthy blood pressure. Could be a little lower. You could be as low as, say, 90/50 and still be fairly normal. What would it mean if those pressures were, say, 150/90? What's wrong with you then? It's called hypertension, high blood pressure. And I'll talk about reasons why in just a minute. Kelsey, how you doing? Caught up? So when you take a blood pressure reading, some of you have gotten to do this in lab already this week, you put the cuff on, you actually pump this up to stop the flow of blood, and then you slowly let the pressure down, and you're able to hear when blood starts to flow back through this closed off brachial artery here. And the highest pressure, the systolic pressure, is the first sound you hear through the stethoscope. The diastolic pressure, the lower pressure, is the last sound you hear through the stethoscope. So you guys will have a chance to do that in lab this week, or next week if you didn't do it this week. So normal blood pressure, about 120 over 80. Hypertension, high blood pressure, maybe something like 140, 150 or so, or higher, over 90 or 100. Why do you think your blood pressure might get too high? What's some of the reasons why blood pressure gets high? Okay, food, like poor diet. What does poor diet lead to in terms of your cardiovascular system? Yeah, cholesterol in your arteries, for example. You can have blocked arteries. If you have stuff built up on the inside of your arteries, you're going to have less space for your blood to flow through, which is going to lead to an increased blood pressure. There's a little, I'm going to have you guys watch this at home rather than have it here, but there's a nice little video on the cardiovascular system. So here's a picture actually of the same artery, both before and after a procedure to remove that stuff built up in the artery. So this is actually called atherosclerosis. Oftentimes it's due to diet, poor diet. You eat too much fatty foods, and that stuff builds up in your arteries. It's carried in your bloodstream, and it's really bad if it actually blocks an artery completely, but even if it's just like this, it's probably going to increase blood pressure. So they can clean that out. They can put a little balloon in there to help force that stuff out of there. An embolism, maybe you've heard of like a pulmonary embolism before or something like that. Embolisms mean that you have something that's floating in your bloodstream that has blocked a blood vessel. So not something stationary like the atherosclerosis, but actually floating. And an air bubble, for example, could be enough to block a blood vessel, or it could be a bit of a blood clot that breaks off, could be bacteria that are built up in the blood, and a number of other things. Anybody know what this is a picture of right here? That's an aneurysm. So aneurysms don't have to happen in the brain. They can happen in the aorta, for example. My mother had an aortic aneurysm that ruptured about 8 years ago or so. Luckily she lived, but a number of people die from those all the time. So an aneurysm is caused by weakening of a blood vessel, and it balloons out like that. It becomes much larger than the surrounding blood vessels, and if it weakens critically, it can actually burst. You can bleed to death fairly quickly. If there are big aneurysms in the brain like this that burst, you may be in trouble. You may not be able to be saved. And unfortunately, they usually don't have symptoms prior to bursting. Hypertension, so here's, if you have any of these problems, atherosclerosis, embolism, aneurysms, here's some of the resulting problems that can come from those. Hypertension, or high blood pressure, defined as greater than 140 over 90, oftentimes due to atherosclerosis, due to poor diet. But it turns out some people have inherited cases of hypertension or high blood pressure. They eat really great, they eat nothing but healthy food, and they still end up with high blood pressure. Why? Well, one possible suspect may be an overactivity of angiotensinogen in your blood. So you might remember from the urinary system last week, angiotensinogen turns into angiotensinogen 2, which constricts the blood vessels and raises blood pressure. So if you have too much of this stuff, it's overactive, you're going to have too much blood constriction, blood vessel constriction, and that could lead to hypertension too, no matter what you eat. Angina, or angina pectoris, that's a general term for like chest pains. If your coronary arteries, the arteries that feed the heart muscle itself, start to become a little bit clogged, and maybe you're out for a run or exercising really hard, you're trying to pump a lot of blood to the heart for all of its job that it's doing, that may cause some pain and chest pain and so forth. You can, I'll show you a picture of this in a minute, but you can sometimes see these blockages with a technique called an angiogram, and you can sometimes correct them with angioplasty. That's where they put those little balloons in there and open up the arteries. You can also have these problems lead to total blockage of a coronary artery, which will cause a heart attack, or myocardial infarction is what it's called, and that's when part of your heart muscle actually dies, or a stroke, when part of your brain actually dies due to having blood vessels blocked to the brain. All right. So I did put a little study guide at the end of this. It's for both the blood vessels and blood as well as the heart part of this chapter. So you can take a look at that for help with preparing and studying for this.